All right, welcome to section 6.2. So in the last section, we talked about the two different types of how we classify those plants as gymnosperms or angiosperms. We talked about the difference in monocots and dicots. And we also kind of went over some of the cells, the components of the cells, and how those cells together form different types of tissues, like the apical, like meristematic tissues, things of that nature, and how those tissues are then used in different components and structures of the plant. So this chapter is going to be the second part of that. We're going to start out with the plant body, discussing our roots and our stems. So the roots are important for absorbing the water and taking up those nutrients, because the nutrients in the water come from the soil. We don't get our nutrients from the air. We get them from the soil by decomposed organic matter. When the soil is formed, some of the minerals that are there and those processes that allow those minerals to become available to the plant. It's also where the water is stored. So when it rains, that water is stored in the soil, and then the plants can then get that water, and in that water is where the nutrients are. So that's how the water and the nutrients get into the plant. If you don't have any roots, you don't have any nutrients. So the roots collect the water and the nutrients that go up the xylem to the plants to perform photosynthesis. Once photosynthesis is performed, it goes down the phloem to give the roots the energy that they need in order to continue their job. They also provide anchor and support. And as I just mentioned, it's where the photosynthates are stored, so that not only do they store that for the roots to be able to continue to grow, but in the event of an emergency, that the roots can send some of those sugars back up the plant, so that other parts of the plant can continue to function. It's amazing how extensive these plant roots can get, and that really all depends on the type of plant, whether it's a monocot or a dicot. So monocots would have a more fibrous root system, again, because remember, it's a stalk that goes straight up, and it needs to have some structure and support. So we mentioned that the roots are going to develop from the radicle. We have two types of roots. We have a tap root and a fibrous root system. So here I mentioned we have a corn plant. On the left is a cereal plant, so this might be like cereal rye. And we have this extensive root system because it has to, it scavenges the soil for the nutrients. So monocots are nutrient heavy. And then we also have this is a carrot plant. And so you can see that this root actually digs down. It actually grows downward, not outward, in order to gain its nutrients. So there's a less volume of soil that can be mined for nutrients and water. So when we have plants like carrots that have a taproot, or maybe like a tomato plant that doesn't have a very extensive root system, we start to apply drip irrigation, because the water is going to move down, and we need drip irrigation and also fertilizer localized placement so that the plant can get all the nutrients that it needs. Whereas on something like a cereal plant, we can do broadcast applications, and we can do kind of like pivot irrigation, or rainfall is going to actually benefit this plant because it has an extensive root system that can mine the soil for water and nutrients. So there are three primary meristematic regions with our roots. Remember, I talked about the root cap. This is going to be kind of like the protection. So the plant is going to, the roots are going to grow, but at the tip of that, where the cells elongate, is like a hard cover so that it protects the cells, kind of like a bumper. A bumper would be a good example of that. So we have this thing that is going to deflect any damage that we might take as we move forward. So, but pertaining to roots. We have the epidermis, the cortex, and then we also have the central vascular cylinder. And remember, our vascular cylinders are composed of vascular cylinders composed of the xylem and the phloem. So we have our cortex, which is just beneath the epidermis. It's going to be where our parenchyma cells are. Then we have this thing called the Casparian Strip. And so this Casparian Strip is kind of like a gate. So it is going to prevent minerals and water from just freely entering into it, kind of like meters how much water and how much nutrients get in. So we don't want to flood the xylem. We want to make sure that we're putting in a consistent amount, and the Casparian Strip is what kind of regulates the movement of water and nutrients into the plant. In the roots, we have the zone of elongation. So there's this area where cells are only going to elongate in. Kind of makes sense, right? The zone of elongation. We have the region of maturation. So after the cells elongate, then they mature and differentiate. So this might be a meristematic tissue. This might be a complex tissue or a permanent tissue. So differentiation, the formation of those tissues, and then we also have root hairs that form. We have the procambium layer. So this procambium is right here that is going to protect the xylem. Here's the primary xylem, and here's the primary phloem. So we have the nutrients come into the plant across the Casparian strip. The xylem carries the nutrients up the plant. The photosynthates are then down the plant through the phloem. We also have the pericycle. Our primary xylem and phloem. So here's our pericycle, primary xylem, primary phloem. Vascular cambium. Some species will have a pith. Here is our epidermis. So this thin layer of cells around the edge of this procambium. And then we have the root hair. So nutrients will come in through the root hair, past the epidermis. In here, they will go across the Casparian strip, enter into the xylem, up to the leaves, photosynthase, back down to the roots. Here we have our root cap. So this is like the helmet or the bumper that is going to protect the rest of this tissue as it continues to grow. So components of the procambium layer, the pericycle, primary xylem and phloem, vascular cambium, and some species have a pith. Next, we have these adventitious roots. These are going to form somewhere other than the radicle. In roots, in corn, you might see these kind of roots that stabilize it. So they kind of like as the plant grows up. Let's say like let's say my neck is the base for the corn plant. You might see some roots that grow out around it that are kind of stabilizing this structure, right? So they're primarily more for support. They also do provide some advantage for taking up nutrients, but they're primarily for support. We can take some of these adventitious roots and use them for cuttings and plants on things like, mine just drew a blank. My mind just drew a blank on which ones we could use those for. They're not chrysanthemums. Straight blank. Moving right along. So here, is a better cross section of that plant. Here we have our, let's go from the outside in. So remember, we have our root hair. We have this epidermis, which is this single layer of cells. We're going to have our cortex. We have nutrients come in from the soil into the root hair and travel through the cells, and it gets to the Casparian Strip. This Casparian Strip is going to regulate how much water and nutrients actually make it into the primary xylem. Next, we will have little areas of phloem that are going to transport some of those nutrients back down to the roots. Oh, and then here's the pericycle. So the pericycle is kind of the epidermis of the cambium layer, for the primary phloem and xylem. So we have like two layers of protection. We've got this epidermis, we've got kind of the cortex and the cork. Then we have the Casparian strip that is going to regulate movement into the xylem, and then this pericycle, which is giving another layer of protection for that organ. Next, we have the stems, and so the stems are going to be scaffolds that develop from three primary tissues. So the protoderm is going to generate the epidermis, so the skin. We have the ground meristem, which is going to generate the cortex. And the procambium, which is going to generate the vascular cambium. So protoderm, derm, epidermis. Ground meristem generates the cortex. Procambium generates the vascular cambium. It's the easy way to kind of remember that. So I mentioned this before. The epidermis is kind of the outer layer. It can be waxy. It's going to protect from desiccation. It's also going to have some influence in gas movement. We have the cortex, which is beneath the epidermis and kind of covers and encircles the vascular tissue and is composed of the different types of tissues or cells. The parenchyma, collenchyma, sclerenchyma, and some other cells. Our vascular system, if you can remember back to that just a few slides ago, the picture of the roots, we have the pericycle, the phloem, which transports photosynthetates from the leaves to the roots. We have the vascular cambium, the xylem, which is going to transport nutrients from the roots to the shoots. Pith rays, and pith. So the pith rays and the pith are going to be kind of species dependent, and so the arrangement of these are going to vary within the three groups that we have. And so here we have our gymnosperms and woody dicot, angiosperms, and perennials. Our herbaceous dicots, so like our beans. And then our monocots are going to be like our grasses. Just to look at kind of the differences, so we have cell tissues from the shoot meristem. Next we have the protophlome and the protoxylem. So proto meaning like the primary, if you can think like a prototype, like this was the first one that we made. This is the primary component. Our secondary tissues, our cambium forms a circle around the stem. The phloem develops towards the outside of the stem, and the xylem is going to develop inward. And then finally, as the secondary tissue grows, it will crush the primary tissue. It needs to have the space. So as the primary tissue kind of matures, the secondary tissue is going to take over, and it's just going to kind of grow outward and take over and crush any of that primary tissue as it makes its way, as it's growing. So in these woody dicots, you might have heard the terms early and late wood. This is what forms those rings in the stem. So when you cut a tree down and you see those rings, that's part of that vascular system. And that growth is in each year are the annual xylem rings as they begin to develop. The phloem is going to get crushed each year and then reformed as new tissue is created. You can removing the bark also removes the phloem, and if we remove the phloem, then the tree is going to weaken or die. So that's why, like, girdling causes a problem. It just basically chokes the plant out, so no nutrients can get to the roots. If no nutrients make it to the roots can't do anything to send any of those nutrients up to the xylem. So if you cut the phloem off, you kill the plant. Here's a better look at this, where we have the rings that are growing through the tree. And as we start to grow outward, we'll have, here's our phloem. We have the cork, and then our vascular cambium, which is going to contain more phloem and xylem. Not that you're really going to need to like go through and say, oh, here's the difference and this is what happens, but just to kind of give you an understanding that there are differences between the different types. So this course is not necessarily concerned with trees, but it is a plant so understanding the simple concepts, we're more interested in plants that we grow for food or for ornamentals. Again, another look. Here we have our primary phloem and our primary xylem. We start to have some pericycle fibers. We have the cortex, parenchyma, and epidermis. So in herbaceous dicots, we have vascular bundles. If you look at this very closely, you can see that it's not just like one tube. It is a bundle of tubes. So we have, because it has to carry so much, if you sent all of these nutrients up or down, in this particular case, it's talking about the phloem. So as the plant, well, first off, you need to have a lot of movement of the nutrients and the water up the plant, right? And only going through one tube, that's going to be very difficult because the roots are taking up water faster than only one tube can transport those nutrients up to the leaves. So we need a bundle of tubes that can produce and give enough nutrients and water to the various plant parts. We'll have that ring of those vascular bundles inside the stem, and the cortex and the pith are going to make up much of that stem. So that's going to be kind of the structure and what gives it its stability, so that those other, and like the protection, insulation almost, if you will, in order to support the vascular system. In monocots, those vascular bundles are scattered throughout the stem. We do not have any continuous cambium or secondary growth of the stem. So once the plant gets out as much as it can, like, it won't continue to grow. Trees will continue to get bigger and bigger and bigger. Plants are typically only going to reach a certain diameter, and that's going to be it. So almost kind of determinate, if you will. But once it gets to be the size, it can only grow so much, and then the rest is just taking up nutrients and sending nutrients to the roots. So we have different types of stems. We have rhizomes, stolons, corms, bulbs, and tubers. Rhizomes, stolons, corms, bulbs, and tubers. List the 5 stem forms other than erect. Rhizomes, stolons, corms, bulbs, and tubers. So rhizomes are going to be horizontal growth below the surface. So take like St. Augustine grass. You're going to find this in a lot of your grasses where you would find a rhizome that is going to grow beneath the surface. So you might cut the plant off, or you might kill the plant with herbicide, but because it has an underground growth, there's plenty of nutrients and storage so that that plant can regrow. Stolons are going to be horizontal growth, but along the ground, so above the ground. And this is how grass spreads. So Bermuda grass, for instance, has rhizomes and stolons. So it can grow underneath the ground and then also along the ground, and it will cover a larger area. Corms are going to be underground, and they are thickened and compressed stems. Next, we have bulbs that are compressed underground stems with many leaves, so like tulips, onions. So when they come up, that they're sending out a lot of leaves. I guess if you think about an onion, it has several layers, but it grows underground. So most of these grow underground, except for the stolons. And then finally, tubers have these enlarged fleshy stems, and when I think of tubers, I'm thinking of potatoes. So that growth does happen underneath, underground. So rhizomes, stolons, corms, bulbs, and tubers. The only one that grows above ground are stolons. The rest of them are below ground. The next important thing is leaves. The leaves are what is going to absorb some of that sunlight, or is going to absorb a lot of that sunlight, and perform photosynthesis and create sugars that are necessary, such as glucose, that are necessary for plant growth. That's where they get their energy from, because they are autotrophic. So we can have different types of morphologies. If you take the lab, you will go over the different types: oblate, star-shaped. We have parallel veins. There are different vein structures, things of that nature. That would be discussed more often in the lab. But in dicots, you're going to find variation in shape, and they typically have a netted pattern. So large veins that branch into smaller veins. Each of those veins both contain phloem and xylem, because how else is anything going to transport? So within the vein, we're also going to have vascular tissues and vascular bundles that are connected to the xylem and the phloem inside the stem. So the stem is going to grow up and then branch out into the leaf. The leaf is going to perform photosynthesis and transport those nutrients and photosynthetates back to the stem, back down the plant. Within the leaves, we have a section of palisade cells that are typically where they photosynthesize. We have mesophyll cells. They also photosynthesize, and that's below the palisade layer. And this is also going to depend on what type of plant it is, on whether or not it's a C3 plant or a C4 plant. So this is just kind of setting up when we go into photosynthesis, that these are the types of cells that are going to be inside the plant, and based on the different photosynthesis type, C3 versus C4, on which type of cells are there. And then finally we have the intercellular space, and this is how water and oxygen and carbon dioxide move within the plant. So we have palisade cells, we have mesophyll cells, and then intracellular space in these leaves. Again, back to the epidermis, it's going to be that single layer of thick cells, one single layer. We have the stomata, which are on the underside of the leaf. Then that's how we are going to take in air and also release air, oxygen come out of the stomata. So carbon dioxide in, oxygen out. And it's surrounded by two guard cells that actually regulate how much it opens and closes, right? So if we have full guard cells and there's plenty of water, it's going to be kind of like balloons. And if you get two full balloons next to each other, those guard cells are closed. And the stomata is beneath it, and so there's no movement in and out. So the guard cells have to open up in order for oxygen and carbon dioxide to either exit or enter the cell. And if it gets to be too dry, signals will go and the guard cells will close and protect that opening from transpiring water or giving off gases that are going to end up almost killing the plant. And they're primary for reducing water loss is pretty much, oh, here we go, regulate the passage of water and gases in and out of the leaf. Here we have the epidermis. We have this set of palisade cells. We have the spongy mesophyll. And then here are our vascular bundles. You can see they're connected here. And so sunlight hits the plant. We have photosynthesis occurring in the palisade cells and the mesophyll, and then transporting those nutrients into the xylem and the phloem. On the underneath of the leaf, we have stomata and the guard cells surrounding that to regulate water and air movement in and out of the plant. There are some different modifications that we're not necessarily focused on, such as thorns and tendrils. Thorns you can think of on a rose bush, and tendrils are kind of like when you see bean plants and vines growing up, let's say a trellis or a wire fence. You know, they kind of grow around that fence, and that's kind of a way to support. But they also have some of those. They also have these epidermis and palisade and spongy mesophyll. It is another way for them to create photosynthase because they don't necessarily have very big leaves, so they have to find another mechanism in order to kind of make sure that they get all the nutrients that they need. We have the leaf blade. We also have the petioles, which are kind of the stem-like, and that is where the leaf is attached to the stem. And then finally we have stipules, which are small leaf-like structures at the base of the petiole. We also have a different number of leaves per petiole. For a simple leaf, we have just one. And then compound being 3 or more, obviously, compound. And that's all going to be determined by the presence or the absence at the base of the petiole. Compound leaves do not have a bud at the base of the petiole as they're attached to a leaflet. In the lab, you'll go over some of these different leaf shapes, leaf types, leaf arrangements. Our buds are undeveloped flowers. We have, they're located at the terminal. Next we have buds, so it's part of the undeveloped shoot or a flower. was kind of located at the terminal of the shoots and the leaf axils. So really where the stem and the leaf are going to start to differentiate, right in between right there, like in between both of those, like where I need another hand is what I need. How about this? I need to quit using that one. All right, so here we go. We have the shoot, and then where the leaf is going to form, the bud is actually like right there. All right. They're going to be mostly meristem cells. You might see them. They might have some protection. They can be vegetative, where they just develop into shoots. They can become flowers or reproductive. Or they can be both. They can develop into shoots or flowers. And they're probably most prominent on deciduous plants when the leaves are absent. So in the springtime, you'll see the bud first. That'll be the first thing that starts to green up and grow, because it is going to develop into the shoot or the flower or the leaf or whichever. Okay? So that's all I got for section 6.2. We have one more section in this chapter. Again, like I said, it's really long. I don't want to have you sitting there watching a 2-hour video, because eventually you would have turned it off and went back, right? So it gives you an opportunity to kind of break this up and not be just tied to your computer. All right? So we'll see you in section 6.3.